Hi everyone, thanks for coming today to the EA Cornish Memorial Lecture. I'd like to, just like to start by acknowledging that we're meeting on the land of the Kaurna people and to pay respects to their leaders past, present and emerging, and also to other Aboriginal leaders on other lands for those online. I'd like to start with can you see that, or is it? Move that out of the way. Just like to start with a brief biography of Edmund Alfred Cornish, who this lecture is named after. He graduated from the University of Melbourne in 1931, and he actually did agricultural studies. And from here, he became interested in rainfall patterns and pasture yields, which is something that's obviously of keen interest to Adelaideans even today, and probably has been forever in the past. So he did that while he was working at the Waite Institute in Adelaide. And from this interest, he decided to study statistics, not just anywhere, but he decided to go over to London and study with R.A. Fisher. And when he came back to the Waite Institute, he was appointed as a statistician. So he transitioned from being agriculturalist to a statistician. In 1940, he was appointed officer in charge of the biometrics section of the Council for Scientific and Industrial Research, which is now the CSIRO. And in 1954, he became the chief of the Division of Mathematical Statistics. And at the same time, the University of Adelaide recognised the need for a Department of Mathematical Statistics, and he became its foundation professor in 1960 to 1965. And during his career, he was a fellow of the Australian Institute of Agricultural Science and Australian Academy of Science. And he was also an honorary fellow of the Royal Academy of Science in the UK. Sorry, this is a bit slow. Sorry. So just a quick summary of his achievements. There was his early work on rainfall patterns and pasture yields, which endured throughout his career. And he's best known for the Cornish-Fisher expansion of quantiles and also experimental designs and analysis of experimental data, including some early work on how to deal with missing data. He's also known for fiducial theory and the multivariate analysis, including the multivariate T distribution. And back in the day when everything was very manual, he promoted the use of computers in statistical work to make calculations much more efficient. And he laid the foundations for the strong tradition of experimental and theoretical statistics in Adelaide. So we'd like to welcome our speaker today. This is Professor Martin Hazelton from the Department of Mathematics and Statistics at the University of Otago in New Zealand. Professor Hazelton's a statistician. He has wide-ranging interests. He's previously worked at the University of Oxford, UCL, University of Western Australia, and now at Massey University in New Zealand. Martin was editor-in-chief of the Australian New Zealand Journal Statistics, and he will be a keynote speaker for the upcoming Australian Statistics Conference in 2025. But you'll be pleased to know that this lecture is an entirely different talk. So thank you, Martin.
Right, it's just the old specified content. Interesting cloud shapes.
Thank you for the introduction. It was a lovely surprise when I received the email inviting me to give this year's EA Cornish Lecture. A lovely surprise and an honour. As you do, I checked the list of previous speakers, and it's a very distinguished crowd indeed. So I thought that I better think pretty carefully about what I'm going to speak about. And I decided upon a topic in spatial statistics, but one that has a very personal connection for me. And the personal connection is that the seminal paper on the particular problem I'm going to look at was written by my own doctoral supervisor, a man called John Bethel. And John is largely responsible for me turning from a mathematician into a statistician. I think he persuaded me by saying statistics is like mathematics, but mathematics plus, which I've become to appreciate more and more as I get older. This talk, in fact, as I say, it's in spatial statistics, and my own doctorate wasn't. I was working on something entirely different with John, but a few years later, I became interested in that side of his work. So, what is this problem of which I am speaking? I say it's a problem in spatial statistics. So the data we're going to be looking at are point patterns. So a point pattern is simply the locations of individuals or events in some spatial region. What's particular about this problem I'm going to be looking at here is that all my points have a mark associated with them or a characteristic associated with them. They're one of two types. So John was originally working in the context of spatial epidemiology, and so I'll actually use lots of his terminology through the talk, where the two types of points are case and control, but as we'll see, any kind of dichotomy for my points is possible. So to illustrate, the figure on the screen is the English Northern County of Cumbria. And what we're looking at here are cases of childhood leukemia between 1969 and whenever it was. It's hidden behind a strange Zoom message. But anyway, so the red crosses, they are the locations of individuals with childhood leukemia. The green circles, they are controls. So they're individuals drawn at random from the population at risk. So children living in Cumbria over the same period. And what I'm going to try and do from these data is answer the question, is there any evidence that the risk of childhood leukemia varies spatially? Now, that in itself would kind of be interesting if we find out there is some spatial variation, but this paper was taken from the seminal article by John Bithell. He chose it for a reason, which is this little blue circle here. That little blue circle is the location of the nuclear power plant at Sellafield. And at the time in the UK, and for 20 or 30 years afterwards, there was considerable concern that living close to a nuclear power plant increased your chances of various kinds of cancer. And so it'd certainly be interesting if from these data, I suddenly discovered that, yes, there seems to be increased risk of childhood leukemia in the vicinity of Sellafield. The second example is another one taken from spatial epidemiology. And it's another one taken from the north of England, in this case, the Chorley Ribble Health District in Lancashire. And what we have here is the red crosses are the locations of individuals diagnosed with cancer of the larynx between 1974 and 1985. The green circles are essentially a control group. They're a slightly odd control group because they're in fact individuals diagnosed with lung cancer over the same period. But the point is that the demographics of those who are diagnosed with lung cancer match pretty well, if you like, the population at risk of getting larynx cancer. So we can view here the green circles as controls, if you like. And again, it's going to be interesting if we find some spatial variation in risk. Are you more likely to get cancer of the larynx in particular areas than others? But again, there's a reason why this particular example has been well studied. That blue circle here is the location of a disused incinerator. And there was some suggestion that this disused incinerator was causing, perhaps, an odd number of local cases of cancer of the larynx. This particular example has been very heavily studied in the problem I'm going to be looking at, estimating the spatial variation of risk, because it's a very difficult one. And people have struggled for a long time to get something that works, if you like, for these data. Now, lest you think that what I'm talking about is entirely aimed at spatial epidemiology, it's not. Here's another data set to which the techniques I'm going to talk about can be applied. It's taken from archaeology. So it's data from a medieval burial ground in Germany. and the circles and crosses here are sites of graves. And one of the things that the archaeologists are interested in is the pattern amongst kinships of burial. So in particular, do you tend to be buried with members of your close family or your extended family? But how would you know? Okay, these are skeletal remains from hundreds of years ago. How could you know if there is some clustering or something similar within kinships? And the answer is one of the things that you can do is you can look at teeth. Teeth are really good in terms of trying to understand relationships from skeletal remains, because things like missing wisdom teeth or missing other teeth, they tend to follow in families. And so the affected data points here, the crosses, are the burial locations of individuals with a particular dental characteristic to do with their wisdom teeth. And so that is a proxy for they come from one particular family group, if you like, and the green circles don't have this characteristic, they come from some other family group. Now, again, I could be interested in, does the probability of having this particular dental characteristic, does it vary over space? But slightly differently to the previous examples, if I did find variation in the probability of having this particular characteristic, it's not necessarily associated with a particular geographical source. Don't get me wrong, it might be there are particular areas of this burial ground that are particularly desirable and that you want your family members to be buried there. But it also could be that there's just clustering. And if I find there's some variation in the probability of having this particular dental characteristic, it might be simply that what I'm seeing is individuals from this family are clustered together. They're more likely to be buried close together. But still, the techniques I'm going to talk about can be applied to this kind of data as well. So John Bithell was interested in how can we explore the geographical variation in risk from case control data. So let's try and put a bit of structure here. So we've got observation relying in some spatial region W, be it an English county or a graveyard or whatever. And John said, okay, there's going to be a density function F on this window W describing the spatial distribution of cases, and a density function G likewise describing the spatial distribution of controls. And then for any particular location in our window, we can define a relative risk function. And that's going to be the focus of this talk, the relative risk function. And that is simply the ratio of the case density to the control density at the given location. The first thing to notice is this is the ratio of densities. As a consequence, it's not going to make any statement about the overall intensity of disease or anything like that. And of course it can't. I've already said that we might be analyzing case control data here. And just because you've got 3 times as many cases as controls or vice versa, that's telling you nothing about the overall prevalence of the disease in question in your population. So I don't care really about the overall numbers of disease. I care about spatial variation. Second thing to note about this is that it's common nowadays not to work with the relative risk function, but with its logarithm. So logarithm is then just the difference between 2 log densities, and that's nicely symmetrized. Now, suppose that in fact there is no spatial variation risk. Suppose in fact that the case density and the control density are the same. Your chances of being found at this particular location X are the same whether you're a case or you are a control. In that case, F would equal G, and my relative risk function would look like 1 over the entire region. And so the log relative risk function would look like 0 over the entire region. So if you like, that's that kind of null model, if you like. Nothing happening, nothing interesting happening. There's just constant risk over the region. But if I find a particular point where the relative risk is bigger than 1, so the log relative risk is bigger than 0, then okay, there's something potentially interesting happening here. Now I need to give a little bit more meat to that, that framework I've just described. So what's the underlying model I'm going to be looking at? Well, the data I'm going to consider to be a marked point pattern. So that means each individual has two things we know about them. One, the spatial location, it's given by X. And second, whether they are a case or a control. So, well, it's 1 for a case and 0 for a control. And this Y is the terminology of spatial stats, a mark. So I've got a marked point process here. If you count up the total number of 1 marks, you get the total number of cases, that's N1. And similarly, N2 is the total number of controls. And I'm going to model the case and control locations as random samples. So I'm going to assume independence. Now, that's probably plausible enough for the cancer examples. Okay? I don't think there's any particular reason why you should be more likely to develop cancer just because somebody close to you has got cancer. Potentially for the graveyard example, it's a bit less plausible in the sense I've already said there might be clustering there. So because one of your kin has been buried at a particular location, maybe you're more likely to get buried close to them. Nevertheless, you could actually handle that kind of situation by saying, oh, but we've got some kind of conditional independence. We could actually think about some latent random field that is somehow describing the overall correlation, spatial correlation in the data, and that given that latent random field, we then do have independence. And that would, for example, be the case if we use some kind of spatial Cox process. Now, I could say more about models, but I'm not going to, because that wasn't what John Bithell was particularly interested in. John Bithell was interested in developing a tool for exploratory data analysis. Basically, he wanted to produce pictures of regions indicating where there might be interesting stuff happening. If you really thought that there was a point source of risk somewhere at a nuclear power plant or at a disused incinerator or whatever, you could build quite sophisticated models. You could test hypotheses in rather advanced ways. That's not what we're going to be doing here. We're going to be taking an exploratory look at things and just saying, okay, here's a first pass. You can apply these techniques to any data set in spatial epidemiology or related fields. And as I said, get a picture. We're hoping the picture tells you something. So the relative risk function is this ratio of case control density.
Go. Jeg er på mute.
Might be a little bit of feedback there. So the case and control, the relative risk function is the ratio of case to control densities. And so we obviously don't know this in practice. We need to estimate them. And clearly a parametric model isn't going to cut the mustard here. You know, human beings don't live on some nice Gaussian surface. Distribution of where we live is highly inhomogeneous and sort of haphazard, if you like, very complex. So we need to estimate these things using non-parametric methods, and the natural choice is to use kernel density estimation. How does kernel density estimation work? Well, I'm sure many of you know, but a quick recap. What you do to form a kernel density estimate is you take each of your data points, you plonk a blob of probability mass on it, and then you do this for all the data points, and those blobs of probability mass begin to combine and form a density surface. If you're wondering about the presence of y and 1 minus y in these formulae here for the case and control, just remind you that y is 1 for cases and y is 0 for controls. And so the top equation just picks out the data points which are cases, and the bottom one just picks out the data points that are controls. So we can use kernel density estimation to estimate the case and control density. Those blobs of probability mass I'm putting on, what they look like is controlled by the choice of kernel function. Usually it is a spherically symmetric bivariate density. Everything that I'm going to do is in fact going to use a bivariate normal. And these kernels are scaled by how dispersed they are. And the scaling parameter is called the bandwidth. If we choose a large bandwidth, we're putting a heavily dispersed kernel on each data point. If we're choosing a small bandwidth, we're putting narrow little kernels on each data point. And then finally, once we've got our kernel density estimates, we can take the ratio of them. We've now got an estimate of this relative risk function. This is a little illustration. Here are the cancer of the larynx cases from Chorley. Let's choose that particular point and plot a kernel function on it. There's another data point, we'll put a kernel function on that one. Another data point, that gets a kernel function too, which combines with the first one, etc., etc., etc. And you can see how the kernel density estimate builds up. And as I said, you can imagine that if the bandwidth was smaller, those kernels would be more peaked. If the bandwidth was larger, those kernel functions would be more dispersed. Now, rather than displaying kernel density estimates using a perspective plot, it's actually, I think, far better to use heat maps, image plots. So here is my estimate of the case density for the Chorley-Ribble data and for the control density. And now I've got density estimates, I can calculate a spatial relative risk function. This, in fact, is on the log scale. I'm always going to display these things on the log scale. So it's, in fact, a log relative risk function. And this is really interesting, okay? I don't know if anybody here is worried about getting cancer of the larynx. Probably never heard of cancer of the larynx. But if you are, what you need to do is you need to move to the east of the Chorley-Ribble region. Because if you look at the scale here, that dark blue is going down to below -6 on the log scale. So that's indicating that if you live in the eastern side of Chorley-Ribble, your chances of getting cancer of the larynx are several hundred times smaller than if you lived in the rest of the region. So remember, what lung cancer is doing is just giving you the background population. So this is comparative, if you like. So, and remember, the lung cancer is meant to be essentially a proxy for population at risk. So yes, so as I said, you'll probably want to move to the east side of the region if you want to be really safe. Conversely, if you're looking at the southwest corner, that's got a rather higher log relative risk, indicating that might be a bit dangerous. You might be a bit more likely to get cancer of the larynx there. And of course, everything I've just said is complete baloney. If you look at those features I've just talked about, this blue feature here and this yellow feature there, they're in areas where there's basically no data, very sparse data. So they're not telling you anything about the true spatial variation in risk. They're just artifacts of my methodology. Could I have expected this? Well, let's do a little bit of theory. There's lots of well-established theory for describing how kernel smoothers work, so how kernel density estimation works. The standard method is to use asymptotics. So we're going to have an asymptotic regimen where the number of data points n is going to become large. The proportion of cases to control is going to remain fixed. And technically, we require the bandwidth that I'm going to use, I assume it's common for both case and control estimates, to go to 0 as the sample size goes to infinity. And this other technical requirement, NH squares, goes to infinity. What this is basically saying is the following, is that kernel density estimation works by local averaging. In order to work for large amounts of data to get consistent estimates, I need to average over smaller and smaller areas, but I need to have an increasingly large number of data points in those small areas to get consistent estimates. So that's all these assumptions are really saying. Couple of definitions, properties of my kernel function, just what its dispersion looks like, and I said this integral of k squared. And we get this formula for the mean value of the log relative risk and formula for the variance of the log relative risk estimate. So let's look at the mean to start with. So we've got a bias term here.
Hvornår er det, vi har møder med 10:30?
So one thing to notice about the bias term is that it depends on the bandwidth. So as the bandwidth becomes larger, as h becomes larger, the bias is going to become greater. But fortunately, if you look at this functional term in the bias, it's telling you that if f and g are pretty similar, that's actually going to be quite small. But let's turn to the variance. The variance, you notice that the bandwidth appears on the bottom. So if you've got a small bandwidth, you get a larger variance, which is kind of inevitable if you think about it, because you're averaging over less points. So you've got more variability. But look at the functional part. I've got 1 over the case density and 1 over the control density. So of course, this is telling me that if in areas of sparse data, in other words, where the underlying densities are very small, the variance is going to explode. And that's exactly what I'm seeing. So the theory is saying, yes, you should have expected it to happen. You should have expected oddities to happen in areas of sparse data. Maybe I can try and fix things by choosing a better bandwidth. I've said you can actually, if you've got a small bandwidth, the variance is going to be big. Maybe you want to choose a bigger bandwidth. A number of people have tried looking at bandwidth selection for this problem. People have tried using various rules of thumb, like Terrell's oversmoother and Scott's isotropic selector. Kelsall and Diggle, Peter Diggle, interestingly enough, is a former EA Cornish Memorial Lecturer himself, developed a cross-validation method specifically for this relative risk problem for selecting the bandwidth. I developed a weighted cross-validation method, and I'll show you how they work in a moment. But before I do, anybody who knows anything about spatial statistics should now be getting a little bit antsy, and I've said nothing about boundary effects. And you see, boundary effects tend to be a bit of a bugbear in spatial statistics. And you can think about why by just considering my kernel density estimates. Suppose I'm a data point that's really close to the edge of my region. I put a kernel function on top of you, and some of that kernel function just disappears off beyond the side of the region. And if I don't do anything about it, that means this data point is having less of an overall impact. That doesn't really seem fair. And in fact, you can show theoretically you get underestimation, quite significant underestimation of things like these densities round the perimeter if you don't do some kind of boundary correction. You can correct for this underestimation, if you like, by using a scaling factor. The scaling factor depends upon the point you're trying to estimate at and the choice of bandwidth in your estimate. And so I could take my case density estimate and divide it by the scaling factor. Likewise for the control. But hang on, I want to calculate the ratio of these two anyway, f hat divided by g hat, and so the scaling factors are just going to disappear. So I don't need to say anything more about edge effects throughout, because rather nicely, they deal with themselves. Anyway, back to, should I have chosen a better bandwidth? Would it have helped? So top left and bottom right are Terrell's oversmoother and Scott's isotropic method for choosing the bandwidth, and they produce pictures pretty much like I just showed you. Peter Diggle, working with a PhD student of his at the time, Kelsall, their method produces a very small bandwidth. The bandwidth is so small that the densities are numerically equal to 0 when you get very sort of in areas of very sparse data, and you end up, 0 divided by 0 for your relative risk. It all breaks down. My weighted least squares cross-validation method chooses a very large bandwidth. I don't know if you can make out the scale, but the scale is much compressed here. So minus 0.2 up to plus 0.3 compared to minus 5 to 1 in other estimates. So basically what my bandwidth is saying is choose a huge bandwidth and smooth everything. But you'll notice that one of the consequences of this is we certainly don't believe there's anything interesting happening close to that industrial incinerator. What to do? Well, we're statisticians, and you've taught stats to a first-year class or whatever. I'm sure you've said something along the lines of, it's not enough to have an estimate of something, you need a measure of precision. you need to give the user an idea of uncertainty. How can we do that in the current setting? Well, again, we can ask Peter Diggle and his former PhD student, and they had the following clever idea. They said, look, imagine going to a particular point X in the region and testing the hypothesis, null hypothesis, H0, the relative risk is 1, or equivalently, the log relative risk is 0. And I do a test somehow. There are various ways of doing it. I've got some Monte Carlo permutation tests. I can also use the asymptotic distribution of my estimator, but I could do a test of that hypothesis. And I could get a p-value. And I could do that at every point in my region, and I get a field of p-values over the region. And I can now say, oh, any points with a p-value less than 0.05, if I'm testing in this case, say, for relative risk greater than 1, those are parts of my region where I really do have evidence that there might be elevated risk. And what I could do is I could take that random field of p-values and I could in fact just select, say, the 5% significance level contour. So imagine this is the surface of p-values. You choose the contour at the 5% level. And any point within that contour is a point at which you would have rejected the null hypothesis that the relative risk is 1. So in other words, you think you might have some meaningful evidence that there is elevated relative risk there. And it's done here. You've got to stare carefully. Here is my tolerance contour right down at the bottom. So this is saying that actually over that region, the only points at which we think the relative risk might actually be elevated is in this tiny little region down here. Although, rather interestingly, that is almost perfectly lies on this disused incinerator. Now, that's all well and good, but it's still not particularly satisfying, because if I'm using these pictures of relative risk, if you like, as a tool for exploratory data analysis, you're still naturally, the eye is drawn to all this other stuff. So maybe I can get better estimates. And one way you can try and get better estimates is say, well, hang on, part of the problem is I'm using a constant amount of smoothing. Why is it a problem? Well, if you've got part of the region with very sparse data, you need a large bandwidth because you need to average over a wide spatial area in order to stabilize an estimate. If, on the other hand, you're looking at part of the region where there's lots of individuals, maybe a town or a city in Cumbria, then you want a much smaller bandwidth so that you can pick out interesting details there. What I've described so far was fixed kernel estimation, but there's something called spatially adaptive kernel estimation, where we allow the bandwidth to vary across space. So here, just for the case density, is a spatially adaptive estimator. Isn't that just what you showed us before? Almost what I showed you before. But if you stare really carefully, the H, the bandwidth here, has itself a tiny I subscript. Because what we're saying is that the bandwidth that gets applied to the ith data point is specific to that data point. And so my aim is going to be to choose small bandwidths at data points where there are lots of data, and to choose large bandwidths in sparse regions. The intuition's there, but this idea was given some theoretical teeth by a bloke called Abramson in the early 80s, who said, look, actually, this works really well if you choose the local bandwidths as following. The bandwidth you apply to the ith data point is equal to some global bandwidth H0 divided by a factor proportional to the square root of the underlying density. And this works exactly as I said it should. If the underlying density is small, so you've got sparse data, I'll get a large bandwidth. If the underlying density is relatively high, I'll get a smaller bandwidth. So this intuitively is doing the right thing. Of course, this would require me to know the true density in order to implement it, but I can just use a pilot. So what's going on here? I can show you what I did last time. Here's a data point in an area of relatively high density, and there's its kernel function. Here's a data point in an area of relatively low density. It gets a more dispersed kernel function. Another data point in a region of high density, it gets one of these peaked tall kernels. Another data point in an area of low density, it gets a far more dispersed kernel. So I'm actually doing what I said we needed to do. I'm applying more smoothing in regions of sparse data. Does it work? Well, there's a few technical tweaks you need to be aware of. For example, do I use the same local bandwidth function for cases and controls? But as long as you do something sensible, basically, you will get an estimator with the following properties. The mean still has a bias term that increases as the bandwidth increases, although again, this functional form is going to be small when f equals g, or approximately g. But look at the variance. The variance no longer has that functional bit that says I'm going to explode when the densities are small. So in fact, I've got approximate variance stabilization here. So this spatially adaptive smoothing seems like a good idea. And so here is the original fixed bandwidth estimator I've been showing you, and here is the log relative risk function using adaptive smoothing. And I will get you to note that the scales are very different. So there's less blue here, and what's more, the blue here is only going down to -3 rather than -6.5. So this is certainly a better picture. And also, you'll notice that the intense yellow area is now more clearly associated with the disused incinerator in the south. I can add tolerance contours as well. Nice tolerance contour saying, okay, I'm pretty sure here that there's elevated relative risk just there. And that's where development stopped for 10 or 15 years. The methods got used, quite widely used, thanks in part to the fact that my former PhD student, Tilman Davies, wrote an R package for it called SPA. So SPA is continually being updated, because Tilman's remains in academia, he's a current colleague of mine. Basically all the methods from this talk are implemented in SPA. A few years later, a guy called Joshua French from UC Denver implemented some of the methods in the SMACPod R package. And there's also a rel risk function in Adrian Bagley's SPATstat that has got less flexibility, but runs about 1,000 times faster than any of these other methods. So as I said, development's then halted for about 10 years. But if we just go back to the picture, I'm still not very happy with this picture. And the reason why I'm not happy with this picture is, as I said, this is meant to be a tool for exploratory data analysis. And you give this to somebody, and they see that picture, and they're still going to be looking at all the different color variations. And there's a danger they're going to do with what I did stupidly earlier with the Chorley Ribble. Oh, there's a blue area there. Maybe that's a very safe area. Oh, they're going to overinterpret. And it's not just that. You think, I've got to be able to do better with estimation, because I know that if there's no data, really, I just want to return a relative risk of 1, okay? If I've got a location where I've got no information, then why would I not return anything other than an estimate of 1? Indeed, I'm going to need some moderately strong evidence before I'm happy with the idea that maybe the relative risk isn't 1 at a particular location. And this suggests that what I might like to do is some kind of shrinkage towards 1 for the relative risk, or equivalently 0 for the log relative risk. So the idea of shrinkage is going to be to shrink my estimates towards the null value, 1 for the relative risk, 0 for the log relative risk, in areas of sparse data, on the grounds that there's then insufficient evidence for those locations to warrant a non-null estimate. Like many good new ideas, you in fact find that your idea is not new at all. I reread the paper by John Bithell, and he in fact talks about exactly this in his paper. Quite briefly, but he talks about it nonetheless. And he basically sort of said, look, my estimates are going to be really unstable in areas of low density, so just add a little bit on the top and the bottom. Now I've changed things slightly in order to make the shrinkage parameter lambda interpretable, but this is the basic idea. I'm adding something at the top and bottom where lambda is a parameter controlling the amount of shrinkage I'm doing. And so you can imagine as lambda becomes very large, these extra shrinkage terms dominate, and that estimate's going to head towards 1. And this is what happens if you apply John Bithel's shrinkage with lambda as 4. The scale is now very much compressed. And interestingly, the only part of the region now where there is this hotspot is around the industrial incinerator. And of course, we've now lost the suggestion that there's particular safety from living on the east side of this region. So shrinkage seems like it's doing something useful here, but I'm still not satisfied. Why am I not satisfied? Well, firstly, intuitively shrinkage makes sense, but I'd like some kind of theoretical justification. I'd like to work in some theoretical framework that says, yes, shrinkage is good to do here, and this is how you should do it. And one of the reasons why I like to do that is because I've got a theoretical framework. It might tell me how to choose the shrinkage parameter lambda. And finally, I still don't like the fact that this picture on the right has all this blotchiness, because somebody might overinterpret it. So in terms of finding a theoretical framework to work in, I can use local likelihood. So what's going on here? Consider a particular point X in your region. And imagine trying to estimate the log relative risk there. And I'm going to assume that in a little neighborhood of X, the log relative risk is flat. Okay, because it's only a little neighborhood. So I'm just going to have a constant estimate in a little neighborhood. Call that value of that estimate in that neighborhood B. So for any point at a location Z in the neighborhood of X, the log relative risk is B. Now here's the thing. If I know a relative risk or a log relative risk, I can also calculate a probability. This is basically, it's just the same as the fact that in, say, logistic regression, you can describe probabilities as log odds or as probabilities. So if I know a relative risk or a log relative risk, I can calculate at least a conditional probability. It's all conditioned upon the sample size. I can calculate a conditional probability. And so here is the probability that a point in this local neighborhood of X is actually a case. And it depends upon my log relative risk B. And because I've got some probabilities, I can now form a Bernoulli likelihood, a binomial kind of likelihood. But remember, this is only meant to hold in a small neighborhood of X. And so what I can do is I can actually say, look, I'll take my data points, the xi, and as they fall further away from X, they're going to contribute less weight to this likelihood. So this is where local likelihood comes in. I am weighting each data point in terms of its distance from the estimation point X. And the weighting is happening using a kernel function. And this is an idea that dates back 40 years to Tibshirani and Hastie. So this is a log local likelihood. If I replace my probability term, or my log probability term, by what I just evaluated further up, I finally get a log relative, a log local likelihood in terms of B. I can maximize the local likelihood, and so I can therefore estimate B. And it turns out that my estimate just looks like the standard kernel estimator. Okay, well done, Martin. You've just shown us how to get the estimator that you used right at the start. Where has this got you? Aha, but I've got a framework now. I've got a local likelihood framework, and I know how to do shrinkage in a likelihood framework. I can take my likelihood, or rather my negative log likelihood in this case, and I can add a penalty term. That's how you can do shrinkage. So the penalty term is going to penalize estimates B that are a lot different from 0. So remember, B is my local log relative risk. The null value is 0, if you like, because the relative risk is 1. And I'm trying to shrink towards 0. What does my penalty term look like? I could have here lambda, which is going to control the amount of shrinkage. K0 is just included to make lambda more interpretable. I could have a penalty term looking like B squared, but I'm going to use the absolute value of B as my penalty term, which basically says I don't want values of B that differ from 0, and I'm going to penalize them using the absolute value of B. And this was exactly the same idea that Tibbs-Rani had for the Lasso. Because you see, the nice thing about the absolute value as a penalty term is it shrinks things to exactly 0. If I'd got something like a an L2 penalty, so I was penalizing B squared, that would shrink things towards 0, but it would never shrink them to precisely 0. But a lovely property of this L1 penalty term, this Lasso kind of penalty term, is that if the shrinkage parameter lambda is sufficiently large, my estimates will get shrunk to precisely 0. And what's unusual in this case, usually when you use the Lasso, right, you've got to calculate your estimates computationally. But actually, I can write down the estimates here. It's a first-year calculus. I can actually write down my estimates of the relative risk function that are obtained by optimizing this penalized negative local likelihood, or log likelihood. And if you stare at some of these estimates, what they're basically saying is that if you get a raw estimate, just f hat over g hat, which is bigger than 1, what you do is you remove something from the top and you add something to the bottom, which is going to shrink it back down towards 1. If you've got an estimate that was less than 1, you would give something to the top and remove something from the bottom. And remember, I said one of the reasons why I wanted to create this theoretical framework is to make me understand the role of lambda. Well, actually, now I can think about lambda in the following way. What the Lasso estimate does is it effectively changes lambda cases at the estimation point x into controls. That's its effect. And so that kind of suggests that maybe lambda is 4 is a kind of rule of thumb on the grounds that, 4 data points is maybe not quite enough evidence to persuade you that something is not 0. Very hand-wavy, but if you think about sort of coin tossing and how many tosses you'd like before you're convinced your coin is dodgy, then maybe 5 is where you kind of get interested. So lambda is 4 is a kind of rule of thumb based upon the amount of evidence, if you like, that you would require to decide your estimate should be non-zero. But you can also do cross-validation. So what you do for cross-validation is you take your entire data set, you hold one data point out, you use the remaining data to estimate a Lasso log relative risk for any given value of lambda. You then use that log relative risk function to estimate the probability that the left out point is either a case or a control, and you form some kind of cross-validation likelihood by doing this. It's frequently the case using this cross-validation method that you get multiple minima. When you do, it's kind of usually a good idea to take the leftmost one. So here's a couple of examples of these cross-validation functions for selecting lambda. And here it is. So here is Bithell shrinkage using my rule of thumb, lambda is 4, and cross-validation. And here's the Lasso. And I was pretty pleased when I got these pictures because it's doing exactly what we want, yeah? It's saying that over most of the region, nothing's happening. You've got flat risk, but there's probably something interesting happening around this disused industrial incinerator. And you've lost all that distracting hodgepodge of blotchiness. Okay, it looks pretty, but is there any real evidence it's giving me some advantages? So I ran some, for example, some simulation studies. Here are some trial relative risk functions or log relative risk functions, one based upon the Chorley-Ribble data, one based upon cases of biliary cirrhosis in the northeast of England, one based upon cases of Campylobacteriosis in the Manawatu in New Zealand. And so I did some simulation studies and I got estimates of errors, if you like. So this is log integrated squared error. So because of the simulation study, of course, I know what the true relative risk function is, or log relative risk function is, so I can work out my integrated squared error. And rather than diving too much into these, I'll just point out that we're tending to find that the shrinkage methods, the Lasso and the Bithell shrinkage, are giving me good estimates. And so rather than spending a lot of time to look about those results, I'll just summarize. Shrinkage helps. In all the methods, I used a common bandwidth, but actually shrinkage might help even more if I used a smaller bandwidth. Because remember, the bandwidth I'm using is having to control or cope with lots of uncertainty in areas of sparse data. But my shrinkage is already doing that. So maybe I'll get even better results if I use smaller bandwidths. And recently, I've just had this year an honours student, Zoe Halls, just say, okay, can you combine the two? Can you apply shrinkage when you're actually using adaptive kernel estimators, so spatially adaptive methods? And we've got even better results through that in a preliminary study. So not only do the pictures look good, but it seems to actually give you smaller errors too. What does it look like applied to my other examples? So here is the childhood leukemia in Cumbria data. Here's my fixed bandwidth estimator. So that's the kind of, if you like, from the original paper, that's what you do. And here is the shrinkage estimator using the Lasso. Again, fixed relative risk across nearly all the region, apart from this highlighted area just on the coast around the nuclear power plant. I should add, by the way, that it's now pretty much established that nuclear power plants aren't giving you cancer. It was just a chance association. But at the time, people got quite excited. What about my graveyard example? So fixed estimator suggests a couple of areas where people from this particular kinship are more likely to be buried. But the Lasso estimator is suggesting it's just at this southern tip of the graveyard. And that might be interesting. I mean, maybe there is something special about the graveyard. Maybe the way that it's developed, this indicates something important to archaeologists. Now, there are various extensions that I could talk about in terms of relative risk. One of the things you might be interested in is how does relative risk change through time? Did some work on this, for example, for foot and mouth disease data in the UK. You might also be interested in comparing relative risk between two time points. So the Campylobacteriosis example I took from the Manawatu, that was kind of interesting because the relative risk looks a bit different before and after intervention for that disease. And so it seems that the intervention not only maybe changed the overall level of disease, but changed the spatial pattern of disease. But rather than talk about those extensions in any detail, I'm just going to conclude by looking at how the methods have evolved. So this top left is the fixed bandwidth estimator. That's what one would have got using methods from John Bithell's original paper. It took 20 plus years for us to think about using adaptive methods of smoothing, and that's what we got with the adaptive smoother. John Bithell's shrinkage estimator is in many ways quite effective, but as I said, it's only quite recently thought about using Lasso shrinkage and got this picture, which I'm still ridiculously proud of, I must say. And so, as I was writing the talk, I thought, you know, actually, there's a few things maybe I've learnt myself about how methodological research develops through this. I mean, any methodological research should start with an interesting problem. If it's not an interesting problem, then certainly for me as a statistician, I'm not sure why I'm trying to develop methods for it. I want to solve real problems, okay? And often your first stab at solving the problem or creating a statistical method or tool to address the problem is a little bit crude. And you'll chip away and you'll make little incremental improvements. And at the time, they may not seem particularly important, but at some point, you're going to look back and say, actually, what we now have is really a lot better than where we started. And certainly, as you're making these incremental improvements, it should always be driven, so far as I'm concerned, by practice. What are the problems with the current methods in practice? What are users having problems with, if you like? What would they like? I'd also like to talk a little bit about the research teaching nexus. You see, it's obviously one of the hallmarks of university is that we've got the research teaching nexus here, and often this said that we're introducing students to our research in the classes. I don't know about any of you guys, but I rarely teach undergraduate students about my research because there's such a big amount of background that they need to understand what I'm doing that it's not easy to include my own research in undergraduate classes. But actually for me, the research teaching nexus works very well the other way around, in that the only reason why I thought about using shrinkage methods here is when I moved to the University of Otago, they said, do you mind doing this paper? It's on modern regression using shrinkage methods. I hadn't spent a lot of time on it, so I'm busy writing up these lectures. And simply as I'm doing this, I'm thinking, hang on. And I think this is actually often true for people in new academic positions. You get given a lecture course, often on something you don't know very much about. And of course, during the interviews, they've said, what could you teach? And you say, oh, I could teach anything. And then they give you this course. Oh my God, I hope they're giving you the course you don't want. But actually, in learning stuff about this new area, it can be a real boon to your research. And of course, you want people to use the stuff. So you've got to release, when you're developing these methods, you've got to release software. So a few acknowledgements to a whole bunch of lovely students I've worked with on this problem. Starts with Elspeth, back when I was at the University of Western Australia. Sarah-Jean Fernando was my first PhD student at Massey. Already mentioned Tillman Davies. Also PhD student at Massey and now my colleague at Otago. Kaya Jones, if anybody asks, I'll tell you something very interesting about Kaya Jones. He was an honours student at Massey. And Zoe Hall's an honours student who I worked with this year at the University of Otago. And also thanks to the Marsden Fund for giving me some money to do some of this stuff. Finally, a big thanks to the guy who started it all, my former supervisor, John Bithell. Thank you.
Thank you, Martin. That was a really fascinating step through spatial analysis. I have to admit, I didn't know anything about the topic, but I'm thinking, I reckon I could use some of that in another application. So has anyone got any questions? Those of you online, you can either unmute yourself or I'll be monitoring the chat. John?
Yeah, this question is stimulated by your choice of K. Now, I usually choose the Epelechnikov kernel. You chose the Gaussian.
Yep.
Yeah. So that means that when you do the approximation, you're doing a mixture of Gaussian distributions, where the mixture is the size of the number of ducks.
Yes.
So an alternative to this whole approach would be actually to choose the number of a mixture and fit a mixture of Gaussians to the distributions rather than fitting the the mixture. Well, what you did.
Absolutely.
And I think it would be interesting to carry out the whole consequences of that, with all the analysis that you provide in your comparison.
Absolutely. So, I mean, in general, mixture models haven't been used as much as one might expect in spatial stats. I had a postdoc who was looking at the use of mixture models a few years back for other problems in spatial stats. I guess, what you can do is you can either be fully parametric and think about a mixture model where the number of components is fixed and finite. Your model will then be misspecified, but you get all the advantages about working in a parametric framework. You can kind of be a bit sort of semi-parametric. You can allow the number of components in your mixture to kind of grow, but not too fast. And then, of course, if you're going to allow your number of components to grow very fast, you might as well just do kernel sort of estimation, as far as I'm concerned. But yes, I mean, you certainly could do that. I agree entirely.
Got a question on boundary effects.
Mhm.
So in the examples, you've got the incinerator right on the county boundary, and you've got the nuclear power plant that's right on the ocean,
Yep.
right? But when you use your kernel, I'm assuming it's symmetric. Does it go over the borders? Because you've got an absence of data there, but is it a real absence? Is it just that you haven't collected it because it's over the border?
So as I mentioned, in terms of boundary effects, you need to correct for them. But fortunately, the correction you'd naturally apply is the same for the case and the control density. And so it cancels out. Having said that, you have not got the same precision in estimation close to the boundary as you have at an interior point. That's pretty easy to show. There's some asymptotics you can do that show that you are asymptotically worse by orders of magnitude, if you like, in the bandwidth near the boundaries. And you're quite right. I mean, the boundaries vary. In some sense, you've got to handle them in every case. But for the Cumbria data, Sellafield really does live on the coast. That's where you build your nuclear power plants, right? Because you've got to cool them. You need lots of seawater. Whereas the industrial incinerator, that's just an administrative boundary.
So I'm assuming there would have been cases on the other side of the boundary. You just haven't.
Yes, but that would require local health authorities to actually talk to each other.
So you don't, you don't think, and you, as you said, you don't think it's biased to results.
As I said, you get less precision, but at least you are getting consistent estimators.
Any other questions?
This is pretty useful stuff in epidemiology and public health in general. And it goes back to John Snell and Broad Street, comfortable and all that stuff. I've seen applications in other areas where the map can be layered. So you have risk factors for that particular location laid on the map. So that's the information provided to give you an idea of what the characteristics of the population are with particular cases of controls.
So certainly, well, actually done that with Sarajini Fernando, one of my former PhD students, where yes, we've got different relative risk factors. For example, for the foot and mouth disease, for farms with different characteristics. So one of the questions you could ask there is, does this profile of spatial risk over the region, does it differ according to what kind of farm that you are using? And certainly from an epidemiological standpoint, that's certainly a very interesting question to ask, yeah.
Got a question online. It says, thank you, Martin. The relative risk estimation of a ratio of 2 PDFs. What about the estimation of 2 empirical CDFs, 0, 1 between case and a control, approximated with our cum sum of PDFs? I'm assuming he means spatially and not temporally.
So was that cumulative?
Yeah.
Yes, so
cumulative density function.
a cumulative distribution function, I assume,
Yep.
in 2 dimensions is a slightly odd thing, okay? I don't know if you ever sort of you can do these things. For example, for positive data, you take the positive orthant and it kind of grows in sort of but it's an odd thing. So
you're saying it's approximated by the cum sum of PDFs.
yeah, I would need to think, but my initial reaction is that anything to do with accumulation in space is not a particularly natural thing, because it's not like, as I said, basically it's because R2 is not ordered, but R1 is. So doing accumulation is a very natural thing to do on the real line. It's a less natural thing to do in space.
Or would you do it in time, for example, if the epidemic was growing?
So you can actually do one-dimensional equivalent of this and ask whether you can get one-dimensional relative risk to it. So you might be able to do something like that.
So do we have any final questions? So was this method used in the COVID pandemic at all? It's not really answered.
No, people have actually done this. It was so there was a Nature paper, for example, on COVID, which uses stuff from the SPIRE library to get some of their pictures, if you like.
Get the spatial distribution of cases.
Well, to compare distributions, if you like. And so one of the things that certainly has occurred to me from this is if you release software, which I say is an important thing to do, you must then be aware that some people will do stuff with your software that you did not intend for them to do. Now, the Nature paper, the way that they'll be using the stuff seemed fine, but certainly, you know, I have from time to time just looked at some of the citations of what people have done. My God, why are you using it for that? I'm not entirely sure I believe it there, but
I don't think we've got any more questions. So thank you, Martin, for coming all the way. So those of you who are coming to the dinner, 7:30 at Eros, will meet you down there. And thank all of you online for joining us today.